Okay, so I'd like to welcome everyone to this latest edition of the DRIIMB webinar series. For those who are not yet familiar with us, DRIIMB is a consortium made up of 4 biological collaborative computational projects. So that's CCP Biosim for biomolecular simulation, CCP-N for solution NMR, CCP4 for macromolecular crystallography, and CCP-EM for electron microscopy. And the consortium is there to promote methods for integration of these different experimental techniques together with modeling and simulation. And we're working on software tools to enable exchange of data and joint modeling. So as part of this, we're running this webinar series to promote the idea of integrative structural biology and to have talks from the leaders in this. And today I'm very happy to welcome Alexandre Bonvin, who has been doing integrative developments for many, many years, particularly from his software package HADDOCK, which I think he's going to talk about a bit today. So Alexandre is a professor of computational structural biology at the Bijvoet Center in Utrecht. And yes, he has a group there looking at integrative methods and software development. So I think this is very much kind of in the spirit of DRIIMB, and so this should be a wonderful webinar. So at that point, I'll shut up and pass over to you, Alexandre. Thank you.
Thanks, Martin, for the kind introduction. And of course, thanks to the organization for the invitations. Always a pleasure to be able to speak. So I'm going to speak today about solving 3D puzzles of biomolecular complexes using both physics and AI-based integrative modeling. So click here, yes. So I'm based in Utrecht, just a little bit of background. So what you see here on the top left is the old academy building in the center of Utrecht. Utrecht is a beautiful city. And the building, the more modern building on the right is the NMR Center. And I've always been doing computational work in the middle of NMR people, basically, and structural biology experimentalists in general. So up there is my office. So if you come to Utrecht and want to visit me, that's where you might find me. And in this building, we host a very large infrastructure for NMR, having both solid and solid-state NMR, 1.2 gigahertz magnets. And we have also been connected to the European grid or European Open Science Cloud these days, having access to more than 100,000 CPU cores distributed around Europe and the world. And this is very important to power the services that we're offering to the community. So what you see here is the center of Utrecht, and my talk is about the social network of proteins. And if you come to the Netherlands, you have to come to Utrecht. It's much better than Amsterdam. Most people know Amsterdam, but what we have in Utrecht that you don't see in Amsterdam is this water level here. You see full of terraces, full of restaurants where a lot of social interactions happen at a microscopic level. And in life, everything, of course, is controlled at the microscopic level by interactions between biomolecules, and in particular, protein-protein interactions. And this has been the focus of our work since many years now, trying to model those to be able to understand those interactions. So looking at the classical approaches, on the right side, you have the experimental world, or a small part of it. And on the left side, you see the more in silico world. So you really only have 3 techniques that can give you full atomistic pictures of biomolecules and their interactions, and it's X-ray crystallography, the oldest of all, nuclear magnetic resonance, actually the first structure was solved about the same time, and cryo-electron microscopy. And next to those, there's a plethora of other methods, and here it is just a small subset, which are going to give you pieces of a puzzle, the 3D puzzle that we're trying to solve. So electron paramagnetic resonance, for example, scattering method, X-ray or neutron scattering, mass spectrometry, FRET experiments. You can run a biochemical gel and learn something about your complex. And the common denominator of all those other methods is that they're providing bits of information, which on their own is not sufficient to basically get a full 3D structure of the complexes that you're interested in. And you have to combine this information and move into the in silico world of computation simulations. And what you see here are the classical method that have been there for a long time. So docking is by now probably close to 50 years old. So it was in, I think, late 70s with Shoshana Bodak and Joël Jonin, where the first docking methods were developed. Threading, homology modeling, provided you have some available structure or homologous structure of your complexes, molecular dynamics. The time for associations here are quite long. So this will be the classical, basically, world of modeling. And of course, as we are all aware, AI has been making a huge contribution here as well. So we have AlphaFold, very quickly, AlphaFold multimers, many different flavors. And you also see in this AI world that experimental data are being integrated. So there is, for example, this paper hidden there, in the back, is about integrating cross-linking data into AI approach. And very recently, basically about a month ago, the EBI, MBL EBI, together with DRIIMB, release also protein complexes in the AlphaFold database. So until now, we had AlphaFold database with a huge number of models in there, but these were all single proteins. And now they have been tackling the problem for interactions. And for now, I think what is deposited there is mainly homomers, not heteromers, but probably at some point in the future, we'll also see the database being populated by AI predictions of complexes. But for today, we'll focus on the more classical approaches. Before going into HADDOCK, basically, I want to take a side step into the AI contribution to structure prediction of biomolecular interactions and navigate the pre and post AlphaFold divide. And this in the context of critical assessment experiments. And this is based on slides provided in part by Mark Lansing and also SG. So Mark Lansing is the assessor in CAPRI, and SG has been assessing the complex category in CASP. And together with Mark, we are running CAPRI actually. So critical assessment, if you're not aware of it, there are two main experiments out there. So there's the critical assessment of structure prediction, CASP, which has been running since '94. It runs typically every 2 years, and there is a prediction season that lasts a couple of months. And actually, the CASP 17 just started on Monday. So if you're interested in this business, you probably still have time to register and participate. It was traditionally focused on protein monomers, but since the couple of last rounds, complexes are also part of it. CAPRI, a little bit younger, 2001, is an experiment that's running on a rolling basis. Basically, whenever we get a target, we are ready to launch a prediction. So we don't have seasons for CAPRI, which means that you can provide targets that have a very tight timeline, because once the paper gets published or the PDB file is released, then of course the competition is dead. So I'm already calling upon all of you here in the audience. If you are structural biologists and you are generating structures of biomolecular complexes, do provide those to these experiments. So you can probably still, if you have things now sitting on your bench, you can probably contact the CASP people and have it part of the current round of CASP, which runs until August. If you have things coming up at a later stage, do contact me to provide targets for CAPRI. These experiments have been critical over the years to catalyze all the development that we have been witnessing in this field. And since 2014, CASP and CAPRI team up in the regular CASP rounds for the prediction of complexes assemblies. So I want to spend a little bit of time to discuss what happened. So this was CASP 14, 2020, and you probably have seen these kind of slides many times. So you have here on the x-axis groups, and there are no names, and on the y-axis in CASP, they have some measure of how good a group is doing. And over the years, for a very long time, the Baker groups and derivative of Baker groups have been doing very well, and from competition to competition, round to round, there are small differences. But this is not the full picture of Cas14 because, as probably you're all aware, there was one more column to add to this graph, and this is AlphaFold 2, basically, which was way above anyone else. The targets in Cas14 were single proteins. At least this assessment is for single proteins. And of course, this was a breakthrough of the year. And if you think, so this was 2020, and they got the Nobel Prize in chemistry in 24 for this work. So this is fantastic. David Baker is part of it, but for the design part, while Deep Rank, Demis Hassabis, and John Jumper for really the structure prediction part. Now in Cas14 and Capri, there were complexes as well. But AlphaFold did not participate to those complexes at the time. So what we did, what Mark did actually, is to take AlphaFold and run all those complexes to see how well you will be doing. So what you see here now are the classical, say, Docker people, and you see they are ranked based on their performance, which is kind of a weighted sum of the quality of the model. So you get a 3 point if you submit a high quality model, which will be within 1 angstrom at the interface from the reference, the crystal structure. You get 2 points if you have a medium quality, and if it's acceptable, it's 1 point. So you see here the best predictors, basically, there were 14 complexes in total, and the best one predicted correctly, acceptable or better, about 9 of those complexes. And most predictions were in the range of acceptable to medium. You see very little red in this plot, basically. Now, if you take a vanilla AlphaFold multimer and you run those complexes, this is what you are getting. So if you look at the triangle, which tells you how many complexes were actually correctly predicted, AlphaFold does not do better than the human. It's also 9. But what's striking is that the quality of the prediction is way better. Now you see a big red bar there. So when it's predicting complexes correctly, it's doing it at high accuracy. So this was a big improvement. But this was Cas14, so we're speaking of 2020. And then we had Cas15, 2022, and there you see that now you start seeing some names appearing, but basically the baseline of AlphaFold multimer is here. So it's in the middle of the plot, and you see that there are many groups that are doing better than the baseline, and all those groups are using AlphaFold or some derivative of it. And using special tricks, they can do slightly better, well, actually quite better. And there are 3 groups that jump out here. And what those people did, so it was Warner, and Warner has been doing a massive sampling. Instead of running 5 or generating 25 models, which is the default for AlphaFold multimer, he was calculating several thousands of models. So basically, increasing the sampling dramatically helped him generate good model. The other team, Paisley Folding, or whatever it's pronounced, they have been basically playing with the multiple sequence alignments. So doing smart multiple sequence alignment also improved the performance. And Vanklovas is a classical Docker/scorer, and he has very good scoring functions based on Voronoi tessellation. He's been using AlphaFold output, but he was very good at scoring, and that's what distinguished them from the remaining of the crowd, basically. And compared to basically baseline AlphaFold, these improvements that have been added to it for the complexes allow to improve the performance of prediction for this round from 30% to almost 90% for the top performing groups. So that's quite impressive. But not all problems are solved. There are still things that don't work very well. And here are just an example. So we're looking at Cas15 and Cas16. So in 16, we saw the appearance of DNA and RNA. They were already there before, but in 16, AlphaFold 3 was there, and AlphaFold 3 claimed they can do the nucleic acid as well. Well, the best model when it comes to protein RNA complexes were from human team who had a lot of expertise in modeling RNA since a long time. None of the AI predictions were making any sense there. On the protein side, you see different things. They are fold-switched system, very difficult to predict. And you see also antibodies and nanobodies being problematic for the prediction, which is not surprising because all those AI methods rely heavily on multiple sequence alignment and coevolution, so conservation or correlated mutations, basically. And in immune complexes, there is no such signals because by definition, those evolve extremely fast. And we can still do much better. So this is Cas16, again, performance of the group. Here, actually, the best predictor for the complexes, so there were 34 complexes, 47 interface. The best predictor is actually also a classical Docker, Kozakov, but he's been using massively AlphaFold methods and also docking methods and good scoring methods, which is what put him there. And you see the prediction of the scorer. But just to give you a few examples, in Cas16, there was also massive fold participating, which is basically coming out of Mark Lansing's lab, which is a massive sampling using AlphaFold, using different models of AlphaFold, doing all kinds of tricks where they generate by default. At that time, they were generating 6,000 models. So it's computationally very demanding. And if you look at those models, and two examples here, this is an antibody peptide complex. This is antibody, or that might be a TCR complex, antibody with some of the proteins, whatever. You see the ranking of the model based on the quality of the model. So the AlphaFold generated, for example, for this guy here, very high quality predictions. For this one, they were also very good predictions, high quality, but the crosses are indicating which was the top ranking model. So in this case, it did select an acceptable quality model, so it's not so bad. And in this case, it generated a bad model, while they were high quality model in the set. Okay, so there is a scoring problem here. So if we were to take the massive fold prediction, if we had the perfect scoring function to identify what is the best model, this is what we could achieve. If we were to take the predictions of all the predictors in CASP and CAPRI in that round, and we had the perfect scoring function that's going to pick out the best model, this is what we could achieve. So there is a big gap between what we could do, provided we solve this scoring problem, and what we are doing. Okay. Which is good news because it means there is still work to be done. So in conclusion, for this small part, there are clearly a big divide between pre and post AlphaFold. Things have changed, and you're all aware of that. But there are still challenges that remain unsolved, and I show you, domain motion, fold switch, immune complexes, and scoring. And scoring, we can clearly do much better. All of this was described in special issues of proteins. And just a short acknowledgement to the CAPRI Management Committee, which I'm leading, and then the CASP is run by John Moult, and actually only 3 people have been running CASP over many years. So again, if you are interested, there's a round starting now at CASP, CAPRI. Do participate, bring new ideas. This is what is catalyzing all the development in the field, or many of the development. So now having convinced you that there is maybe still room for more classical approach because not all problems are solved, I'm going to switch back to integrative modeling with HADDOCK. So HADDOCK has been there since more than 20 years, and it's developed as an integrative modeling platform. It started from an NMR problem where we started using NMR titration experiments, basically NMR experiments that are telling you where something is binding, but not how. So we know which residues are important or involved in a binding, but we don't know which contact they are making. And that was basically the trigger for the idea to implement or to use this information where you know something is important, but you don't know what it's doing, into highly ambiguous interaction restraints that are basically targeting multiple residues on the partner protein. So it's basically a distance between many points, thousands of points that we're calculating. And we put those distance between the molecule to draw the molecule together. So it started from NMR, and you see NMR has many contributions in terms of studying complexes. Many experiments provide you bits of information. Mutagenesis is valuable. Cross-linking, linked to mass spectrometry, extremely valuable. Very little sample quantities needed. HD exchange. Bioinformatics, of course, you can try to predict contacts. Coevolution has been there since a long time before it was used in AI and AlphaFold methods. And we have shown that this is also very useful to model complexes, of course. And over the years, we've also added support for cryo-electron microscopy. So I think we are covering most of the CCP parts that are represented in this initiative. So what kind of molecule can we deal with? We started with proteins, which was the question we were trying to answer at the time in the NMR lab. And since then, we have been expanding to many different types of molecules. So proteins, small peptides. Peptides are not different than proteins. They're only different in the fact that their flexibility and conformational variability brings challenge to the modeling process. Nucleic acids, also pretty early on. Glycans is something that we have added support more recently, especially during COVID. There was a lot of demand for glycans and glycosylated proteins. Small molecule docking has always been there in HADDOCK since the early days, but the problem is that you need to generate conformations, and you need to generate topologies and parameters for those. The server has been handling that under the hood. And we also added more recently shapes, which are basically an arbitrary collection of what we would call beads. Those have no energetics with the system. We'll typically fix those in space, but they allow you to represent all kinds of objects. So you see here 2 planes. This would be a very crude representation of a membrane, where you could say, okay, this is the position of the phosphate atom of my lipids. And you might think that, well, if I'm docking 2 membrane proteins interacting in the membrane, they should move inside these 2 planes. And if you have a membrane protein interacting with soluble protein, one of them will be inside the 2 planes and the other one should be outside. So this is providing you basically topological information that you can transform into restraints through modeling the process. And this here could be a reconstruction from small angle scattering data. You could do a reconstruction, a bead reconstruction for low resolution cryo-EM, and then the games become to dock the molecule into those shapes. And we also have coarse graining in place in the server. In terms of restraints type over the years, so everything started with distances and these highly ambiguous distances that allow you to encode basically anything in terms of interactions. A lot of NMR data have been added over the years because, well, being part of a big NMR lab, this was a natural thing to do. We incorporated cryo-electron microscopy data so we can refine against cryo-EM map, and for the docking part itself, we extract from cryo-EM map centroid positions where the likely position of the molecule might be and define restraints to those. Symmetry, very important when you're dealing with symmetrical systems, and non-crystallographic symmetry is something which is familiar to the X-ray people, but this allows us to basically define relationship between molecules that should have similar conformations. Now, the classical docking protocol in HADDOCK consists of 3 stages. In the first stage, we're going to treat the molecule as rock solid, and they come together by energy minimization. So the starting point is the molecule separated in space, randomly rotated, and then we turn on the minimization, driven by the data that we put in, and driven by electrostatic and van der Waals energies. This is typically a fast process where it's a question of seconds. We're going to sample 10, 100,000 models in that range. And we will do a first selection of a few hundred, and then those go into the second stage, which is a flexible refinement stage where only the interface gets flexibility, both at the sidechain and backbone level. And the remaining of the structure is rigid, but can move in space. We're using torsion angle dynamics for that. And at the end, we used to put a layer of water around those solutions and do a very short molecular dynamics. We're speaking of 10, 20 picoseconds, so it's nothing, it's really very small, just to optimize a little bit the energetics. These days, we just do minimization at the end. So this is how it looks like. Not sure how well the movie will stream, but you see on the left side this rigid body minimization process, and the residues that you see as spheres are the ones that were in this case identified by NMR to drive the interaction. So they are drawing the molecule together based on these ambiguous restraints that we define. In the central panel, you see the torsion angle molecular dynamics. So it's a simulated annealing protocol. We start at high temperature, cool down the system. Dynamics first along the side chain and then along the backbone. So things are, you see now the side chain will start to move. And at some point you see a loop here which will flip over. It's a small binding induced conformational changes. This kind of protocol will never fold an alpha helix from a random peptide or things like that. So the amount of conformational changes that we can deal with is very limited. You might sample conformations prior to docking with other methods and then provide an ensemble of conformation as starting point for the docking. That's often, if you are dealing with large conformational changes, a more efficient way of dealing with that. Now, how do we, so when we do the calculation, we have a kind of a classical force field, van der Waals energies, electrostatic, bond angles, dihedral impropers. And when we score, we only use a subset of these energetic terms. So they are basically three colors that you see here. So we have slightly different scoring function at different stage of the protocol. So this is a simple linear function which has very round and weight, as you see. This was derived 25 years ago based on a few complexes and chemical intuition, and this has survived 25 years of optimization. I'm not saying it's perfect, it has its flaws, it has this problem, but it's robust, and that's very important. And more importantly, we use it for mixture of complexes, protein, nucleic acids, only for small molecule where we might change some terms at some stages. So what are those terms? So the green one is basically the intermolecular electrostatic and van der Waals energies between the molecules, and the electrostatic is scaled by 20%. The blue one is an empirical dissolution energy term. So this term is not present during the calculation. We only use it for scoring. And it basically measures the bonus or the price that you have to pay to dissolvate an interface. If you bury hydrophobic surfaces, usually you win energy. It's an entropic term because the water is released from this hydrophobic surface and goes happily into bulk solvent. If you dissolvate charge residues, you usually have to pay a price because this costs energy. And the last term, which we call ambiguous interaction restraints, is basically representing the data that we put into the modeling process. So HADDOCK has become quite popular because since 2008, we have been offering it as a web portal, which makes it use easy. And it also provides a computational resource to do this kind of computing. And you see here the current version of the portal. So we have passed more than 80,000 registered users, slowly getting to 1 million served runs since the first days of HADDOCK. And we can do that because we have access to these high throughput computing resources, grid computing, if you want to call it. With the support of EGI, which is basically the federation of all the national sites that are providing the resources. So jobs are distributed, and the server itself distributes probably about 20 million jobs a year to those resources, mainly in Europe, but some jobs end up in Latin America, some used to go to the US, some were even running in China. And quite a fraction of those actually are running in the UK at RAL. So we are very thankful to have these resources. This is a current overview of our user base. So it has been growing quite fast, and it's amazing to me to see how many countries are represented. In some countries it's maybe only one user, but still 188 countries represented in our user base. And if you think that in the United Nations we have about 196 countries or something like that. So I would have expected with the rise of AI that we would have seen a flattening in terms of usage. This is not there, so we are speaking of 2020, so AlphaFold, CASP was 2020, somewhere here. We're still basically pretty much in an exponential growth phase. So this was to us a surprise, but maybe people are using also a tool. So you can do docking with it, you can also do refinement. So maybe it's also used quite a bit in refining AI models. The same is true of registration. So this starts in 2024. What you don't see is the COVID period, where basically we had a doubling, tripling of usage during COVID. Everyone started doing modeling because you were locked at home and couldn't do experiments. And we see again since last year, again, almost a doubling in usage. Where this is coming from, I don't know, actually. It's used more and more in education as well. We published quite a few papers last year about antibody and antigen, well, nanobody modeling, so maybe that's the thing. And again, the last month, this is March, big jump again. We'll see what April brings. together with more users. So these are monthly registration. We also see that the server is processing way more submissions. So last month was a peak close to 20,000 docking runs submitted by users. And one docking run translates into several hundred or several thousand small computational jobs that are distributed around this grid of resources. Majority of your users these days is from Asia, actually, then Europe aggregated, North America, and then the other regions. So what I presented now has been basically the classical HADDOCK method, which has existed in different versions and iterations and improvements since 2003. And over the last 5 years, in the context of BioXcel. BioXcel is the Center of Excellence for Computational Biomolecular Research. It's a European project, which is now actually funded by EuroHPC, Joint Research Unit. We have been breaking HADDOCK in 2 pieces, and this has been given birth to HADDOCK 3, which is a modular version of HADDOCK. And you see some of the people here who have been doing some of the major work. So we took this static workflow, which is also what the server is running these days, where you can change many parameters. So you can change the length of the protocol, the number of empty steps that you are doing. You can change the scoring functions, the way you score, but you are rigid in terms of the workflow that you are running. So this is the HADDOCK 2 version, basically. And now we're basically breaking the code into small modules. So we have a catalog of independent modules. There are a few dependencies, though. And then you can use those modules to mix and match and build your own workflows, do only the things that you need, or build much more complex workflow. This is a very nice flexibility to have. And this is the current, so the latest stable release is March 26. So you have topology modules, so you need to build the system first. This is going to build any missing atom. You have a lot of analysis modules that we have added. A lot of them are related to Capri as well, but also clustering methods. You can do nice contact map analysis. You can do alanine scanning automatic of your interface using HADDOCK. We have the sampling module. So this is the classical rigid body energy minimization one. Third-party software like DRIIMB, CheatDock will probably be, that's a genetic algorithm-based docking, which might reappear. We have the refinement module, so flexible refinement, what I showed you, energy minimization, short molecular dynamics with water or not. And we have also added OpenMM as a more molecular dynamics engine to optimize better the complexes, but this cannot handle the restraints. We have modules for scoring only if you're just interested in adding a score to a model that you generated with some other methods, and there are a number of command line interfaces. The definition of the workflow is quite simple, so it's a YAML format or TOML format actually, where you define the modules that you need. So this is a workflow that will reproduce what the HADDOCK server is doing, and you just need to define the parameters that you want to change. You don't need to touch the default one. So that's quite a simple input in principle. What we also added to HADDOCK 3 is basically a graphical analysis of the results. So it's providing HTML tables that present you the same information that the server is giving you. So before, if you are running a local version of HADDOCK, you had to do a lot of manual work to analyze the data. The server was doing that for you and presenting you the results in a web page. Now, when you run HADDOCK 3 command line, you get the same output as the server, basically, and you have this interactive graph where you can zoom in, zoom out, turn on cluster, turn off cluster. And very recent, so this came out in bioRxiv yesterday, I think, or Monday. We have put back coarse graining into HADDOCK 3, so we added the Martini force field into HADDOCK 3, and we built new modules for those. They were not there in the slide I was showing you because we don't have an official release, but if you take the latest version on GitHub, you will get it. So we have a module which can basically convert an all-atom model into a Martini model, and then you will do your docking workflow at a coarse grain level. And then we have a module that basically converts back the coarse grain model into an all-atom model. And if you want to look at the detail of that, check up the bioRxiv paper. So what can you do with all of that? Let me start with a recent work, which is an NMR-based application. And this is work which was done in collaboration with Beate Kragelung. She's at the University of Copenhagen, and she's the champion of looking at disordered system and disordered proteins. And our PhD student Kira came to Utrecht to do some modeling because they were running into problem to study this system. So we're looking at the SH protein from a mumps virus, and you already see a model that came out of this. So from experimental data, simply from gel already, but also from mass spectrometry, it was clear that this protein is forming a pentamer. And it's actually a membrane protein. Again, you see the model, which is the outcome of what we did. NMR tells you, NMR in the HPC micelle tells you that the protein is folded into an alpha helix. Since it's a symmetrical pentamer, you only see one set of signal by NMR. You can also do CD that shows you it's alpha helical. And from the analysis of chemical sheet data, but also from relaxation data that tells you things about dynamics, you can see where the helix is in this. So there is a disordered region on the N terminus and a bit of disorder in the C terminus, but the central part is actually ordered. Now, if you give the sequence of this protein to AlphaFold, it models it as a straight helix, which you see here with quite high confidence. Okay, so it's everything is above 70, and a large fraction of it is above 90. We also try to do the modeling of the complex, the pentamer directly, since we knew it was a pentamer. So there, AlphaFold is less confident about the structure. It still modeled the entire thing as alpha helical. And if you are used at looking at structure and you start looking at those models, and I think that's an important message here, if you do any kind of modeling, don't trust numbers, look at structure. That's really very important. Now, if you look at structures, these are basically the top 5 ranked model from AlphaFold 3, and 2 gives you the same thing. What is strange here, you see polar residues. So there's a tyrosine here, there is a tryptophan here, there's a threonine somewhere, and all those polar residues are actually facing the membrane region. And the inside of the pentamer is basically hydrophobic. Now, in a membrane, this does not make sense. Okay. So this was where we were standing when Kira basically started, and then we decided, okay, let's try to use HADDOCK so we know which region is alpha helical from the NMR data. So we're going to model that as a perfect helix. And then we use HADDOCK to do a symmetrical C5 docking. So we impose symmetry restraints, and we use simply center of mass restraints to bring the molecule together. We don't have information about the binding site because it's a symmetrical system. You don't get this chemical shift perturbation. And here you see some of the cluster that HADDOCK gives you. And the good thing is you see directly that basically this is physics-based modeling and not a physics boost the polar residue in the inside of the pentamer. And if you analyze more some of those models, you realize there's actually a channel through this protein, through this pentamer, and this channel from the experimental work that was done in Copenhagen was shown to actually conduct chloride. And now this makes sense. Now it also makes sense that you have these tyrosine residues here, and you see the threonine here in the center of the channel for the conductivity of the system. We now have a model. We can generate hypotheses from there. So seeing this tyrosine in the center, we decided to mutate it because it must, if it's functional, it must do something. So now you see a patch clamp experiment where you measure the conductance of the channel. So this will be the wild type sequence. So you see a nice current here. And this is when you mutate this tyrosine to an alanine in the middle of the channel, you lose the conductivity basically. Tryptophan is also found in the channel. The tryptophan has a different origin, so it's very important also for basically if you mutate it, you lose conductivity, but you don't lose it because the channel is not functional, but you lose it, the power of NMR, because it's not forming alpha helices anymore. So basically you have disrupted the pentamer by doing this mutation. So that's an important residue to stabilize the pentamer. And then you can even drug these systems. So this is one molecule, which was, so the work was done in Copenhagen here, where you can block the conductance of the channel by adding this drug. You can also monitor the binding by NMR. So this established basically this protein, this SH protein, as a possible drug target to derived antiviral compounds. And here, HADDOCK modeling, physics-based modeling was key to generate models that can basically generate hypotheses and more plausible model than what AI was giving. By the way, these days you also say in AlphaFold 3 you can add lipids to your predictions, and it helps usually when you do membrane proteins. We try to play that trick in this pentamer. It was still generating basically the inside out model with the polar residues on the outside. Okay, so now looking at the time, I'm going to go quickly over the antibody antigen modeling and probably skip the scoring part. So, with the advent of all these AI methods, and we have millions of sequences of antibodies these days, so there is no excuse not to be able to model those. But we wanted to see basically what are the limits of the modeling of antibody complexes using AI. And for this, teaming up with a team in the UK actually, these are people, and Charlotte Dean was part of this effort as well. This is ex-Scientia team. At the time, they were very interested in antibodies. So we selected about 80 complexes that had not been seen by any of the AI methods that were trained at the time. It's very important to have very stringent selection, otherwise you will have, your model might have seen your complex before, might have seen something that resembles heavily your complex, and then you're not doing a blind prediction anymore. And we put AlphaFold Immune Builder, that's an AI model from Charlotte Dean's which is very fast, actually very nice to use, and IgFold is coming from Jeff Gray. So we put them to the test. We did a very fast HADDOCK protocol, so this is HADDOCK 3, so very limited sampling. We generate only about 200 models at the rigid body stage and refined only 50 of those. You see 196 and 48. We had 96 core nodes at the time, so 48 allows you to run two on the same node. And here, of course, we assume that we have some knowledge on the binding side, on the antigen side, and for the antibody, we can use the CDR. We know which loops are involved in the binding to target the binding. So this is not a protocol if you have no idea where your antibody is talking to. And you see the performance if you give the true interface, so you have the exact definition of where the binding takes place, not how, but where. If you have a more loose interface definition, something that will come from maybe NMR or mass spectrometry, you see we take a hit in performance. And the two column is if you take a single model coming off AlphaFold or Immune Builder, but if you merge all the models from all the methods, so their sampling becomes a bit better, that's where you get the best performance. So it's worth running several models and combining the predictions. And this is the performance of AlphaFold on this same set of 80 complexes, which is not surprising because there is no coevolution signal. Where we fail, it's mainly linked to the H3 loop in antibodies, which is the longest loop involved in the binding, which has also more variability in sequence and usually undergoes conformational changes upon binding. So these are basically the challenges. Conformational changes are challenges when it comes to modeling complexes together with the scoring. So in our data set, we selected about 50 entries which had no sequence redundancy at the binding site with anything that AlphaFold had been seeing before. And we generated a model for all of those. And you see here the problems. So there are 6 loops in antibodies. 5 of them you can model with quite high accuracy, but the H3 one is the problematic one. The good thing about, and you can try to play also, you might have heard about these methods to increase the sampling of AlphaFold and related methods. So you can do increase the number of seeds, play with the multiple sequence alignments, do dropout in your network. We even tried biomu at the time, but this was after the review reports were in. This was not helping us in this case. The good thing about AlphaFold for this particular case is that if you calculate what is the predicted PLDT of the H3 loop only, there seems to be a correlation between the predicted quality of the loop and actually the RMSD from the bond form. So this is nice because it tells us that looking at the PLDT of the H3 loop, the cases where we have where we are below 18 PLDT, these are the cases where we can expect larger conformational changes. So we have to do something about those. So around that time, another AI method was published coming from MIT, AlphaFlow, which is a flow matching AI approach, and it can be used to sample conformation. So this was before biomu was out. And the idea that Xia Tong had at the time is basically to start using AlphaFlow to sample H3 conformations. And this is an example of what you get with AlphaFlow. So you see a sampling of H3 conformation. The blue one is the model generated by AlphaFold, and the orange one or the yellow one is the model, the crystal structure of the complex. And you see that AlphaFlow is able to generate conformation that resemble quite closely the complex. The problem is it has no good scoring function, so you don't know which model you should use. So we generated 1,000 models for each loop, but we don't know which one is the good one. But you can see that the distribution of model is much better after running this AlphaFlow. So how are we going about modeling those? So we do this AlphaFlow sampling of the loops, and then we do clustering. We request 20 clusters, and we take one model, the centroid of the cluster, and we give those 20 models as an ensemble to HADDOCK to do our targeted docking. And if you do that, you can see that you can improve a lot. So the orange is what you will be getting if you take straight the AlphaFold prediction, and the blue and green one, blue and green means blue is acceptable quality, green is medium quality, is what you are getting if you're doing this AlphaFold based approach, and you see that we are increasing indeed the success rate. But if you have much less well-defined interfaces, which is the right side, you see that the performance is still limited and our scoring is still not stellar. So again, immune complexes remain very challenging. So AlphaFold 3 has improved things, and they report that in their paper. It's at the cost of a massive sampling again. It's hidden in a supplementary material. On the data set that I show you, AlphaFold 3 only help in 1 out of 9 cases. So again, it has not solved all the problems. So in conclusions, AlphaFold and related AI methods have changed the way of we can study antibody, nanobody complexes, but they still are faced with challenges in H3 because of conformational changes. So exploring widely this H3 is the way to go. And the same holds for nanobodies. I didn't speak about that, but there's a preprint on Bar Archive if you want to look at it. And this is the paper about the AlphaFlow part. So for now, I think there's still need for more classical integrative modeling approach next, or combined with the AI approaches. So this brings me almost to the end. So I will run through a few last slides. Because I started showing you that scoring is still a big problem in CASP and Capri, and the AI have not solved those. So we started quite some time ago to start developing AI methods to score complexes. We started by implementing a 3D computational network where we basically treat the interface of a complex as a 3D image. This gave rise to DRIIMB. But with 3D image, you have to learn to recognize them from different directions. So for training, you have to do rotation. So it's not rotation invariant. So we moved from a 3D to a 2D representation as graph neural network, where we represent the interface as nodes connected by edges if they are close by, and we distinguish the intramolecular nodes from the intermolecular contact. So we train a network to do this classification. So this gave rise to DRIIMB GNN from graph neural network, which was trained basically to predict the quality of the complex. The key feature in this is still the position scoring, a specific scoring matrix for which you need multiple sequence alignment, which might be a limitation again for immune complexes. So we've been looking at large language model, and we replaced the multiple sequence alignment by ESM2 embeddings. And this gives rise to DRIIMB GNN ESM, which is also out there. And basically, you don't lose in performance, and it's much faster because you don't need to calculate multiple sequence alignment. And now we have also been working on an antibody-specific version, the work of Xia Tong and Ilaria. Ilaria was a master student where we generated millions of models for about 1,500 complexes, extracted a test set, which is not used in training for validation. And we added module to our DRIIMB. So we defined, for example, the loops of the antibodies as features in a model. We added contact. We added atom areas. So Voronoi tessellation is used in there. We added energetics to the model, orientation, and we train an equivariant graph neural network basically to make the predictions. And this is the results of this. So we have different models. So these are all different models from DRIIMB AB. HADDOCK scoring is there as well. FTDMP is from Penclova's group, who has been doing very well in scoring in CASP and Capri. So this is the Voronoi-based approach, and AlphaFold 3 is there as well as the blue line. So you see that the best model, which is this one here, is basically outperforming all the others. What is interesting also, is the problem solved? No, it's not. We are correlating here the ranks of AlphaFold versus the quality of the models measured by DocQ. So AlphaFold has basically no correlation between rank and values. So everything is around 0. DeepRank is doing better. So for some complexes, these are all the complexes that we use. Some complexes, we have very strong correlation between the score reported by DeepRank and the real quality. For some, it's anti-correlated, problematic, so the problem is not solved. Those special complexes, we try to look into this, but it's not really clear. And for some, there's no correlation. So again, it's not a solved problem. This is available on bioRxiv as well, and it should appear very soon in print in a journal near you. So let me wrap up. So I'm very proud to be here and talk, about integrative modeling and HADDOCK, something which has been going on for by now almost 25 years in the lab. So despite all the booming AI and all the new methods that have appeared, we are still in business, which is quite amazing, I must say, because AI is really doing fantastic things. But for now, there is still need for more classical integrative modeling approach. So it's not like one should replace the other. So I think it's a combination. So we have to embrace AI, but we also have not to forget about what physics gives you. And I show you the example of the viral pentamer protein as a good example for that. And you just discovered maybe the new version of HADDOCK, HADDOCK 3. We are also working on a web server for that. So let me thank the people. So this is a very recent picture of the group from last week. The weather is quite nice in the Netherlands this week, so we had a barbecue. Funding over the years from different national and European projects, and these days BioXcel has been really important in funding software development, which is not something easy to fund. So thanks to all the team. Thanks to all the developers over the years. We celebrated 20 years of HADDOCK in 2003 at a good party and a bit of science. And thank you very much for your attention. And while waiting for questions, a bit of advertisement. BioXcel is organizing a very nice conference in September this year in Brno. Very nice location. So look it up. It's about advances in biomolecular simulations. So it goes from multi-scale modeling, molecular dynamics, free energy, integrative modeling, AI, drug design. Yep.
That's brilliant. Thank you so much, Alexandre. And I'm very happy as well if you send it to me to advertise that wonderful looking meeting on the CCP Biosim website.
Yes.
So we'll send it round on our mailing list. If you send it to me, that would be brilliant. So you do have questions. So we have questions from Rivka. Hi, Rivka. Lovely to see you. So Rivka asks, she says, I've read about the kind of targets that CASP and Capri are seeking, but would like to know more about how much experimental data is ultimately needed to verify the predictions. For example, if we have a DRIIMB structure of a dynamic multimer with variable resolution and variable occupancy for different conformations, etc., is that still useful? And what about lower resolution data? For example, pulse EPR distances measured at various points between a homodimer that still has quite a dodgy alpha fold prediction. So what would you say, Alexandre?
So basically, we need to assess the quality of the predictions. We need to have some ground truth. So far, we have been thinking, EPR distance are interesting. Cross-linking is giving a lot of information these days. But then we have to rethink the way we measure the quality, because you cannot say that the model is good and bad. We could only measure the quality in reproducing or explaining the data. So we have not yet moved in that direction. In terms of type of targets, anything is nice. The first thing we do is typically to run AlphaFold. You know, if it's a classical protein-protein complex and AlphaFold nails it, it might not be very challenging because I think everyone in the community will start by running AlphaFold on those. So if we look at what we have been running over the last couple of years and the focus of CASP this year, so immune complexes are the tough ones for many reasons that I showed during my talk as well. But you know, flexible systems are interesting, where you have so one of the last round of Capri was prediction of several conformation sensitive antibodies. So antibodies that will recognize specific conformation of the same protein. Again, a trimeric viral protein. So these are challenges. So if I hear, you know, cryo-EM, different states, this will be very interesting, interesting to look at. Low resolution cryo-EM, again, you need to be able to get some information of the global arrangement, but then we could not measure contact. So I think there's but we hope, you know, cryo-ET should probably give us a lot of interesting targets out there, where the resolution might not be from stellar, but still it could be interesting to look at those. But conformational changes are a challenge. So anything which has massive large conformational changes, but also glycan peptides. What Capri typically doesn't deal with are small molecule. They are over competition, so over experiments for that.
Excellent, thank you. So Charles says, very interesting talk. I was wondering, what is the success rate for prediction of the interaction between an epitope and CDRH3?
So the success rate, so if you have an idea of your epitope, you know, you get it from NMR, HD exchange, mass spectrometry, whatever, the success rate in HADDOCK, in what we're getting, we get up to 80% success rate on the benchmark of these 80 complexes. So again, if you have you might get slightly lower if your definition is very loose. So if you have, you know that it's this region in the domain which is binding, then it drops around 60 probably. But it's still better than the performance that AlphaFold will give you. So of course, I think that's the power of HADDOCK. If you have good data, we can drive really the modeling, and that makes it. But if you have very loose, very little data, so if we don't know at all where the epitope is, it's still a very hard problem. And this is typically what's happening in a Capri. In the last rounds of Capri, we had antibody antigen complexes where you have no information. And it's very hard to predict also epitope. So epitope prediction is also a field which is not solved, because these are typically not, the conserved region of the protein. It can be anything. So it's still a tough problem.
Excellent, thank you. So Miriam has a little bit of a problem with her docking. So she says, so her HADDOCK scoring didn't go very well. And she said, what else would you recommend for a protein peptide complex?
Yeah, peptides, again, that's well, I guess the question is, how do you know that it didn't go very well? So maybe you had very good data to bias the modeling. So for protein peptide, it's not very simple, because there is, first of all, I think, you have AlphaFold rank and some of these other AI methods that seems to be doing quite well in some cases, but there is not a very large number of protein peptide complexes to train AI model on those. And that's a problematic thing. The other thing that you should realize, and I didn't speak at all about binding affinity, but when we speak of scoring is not a prediction of binding affinity. Okay, so if you're looking at docking different peptides and you want to know which one binds better, the scores that we get in docking are not meant to do that. When you do docking, you assume that two proteins are interacting or two molecules are interacting, and the score is there to try to tell you which model is the most likely correct one, or the better one, but it's not telling you which one binds stronger. And that's a very different problem. We've worked on that as well, but that's a hard problem. And what most docking method will not give you, if you give them two proteins that are not interacting, any classical docking method will give you a beautiful complex and a beautiful model of it, but they will never tell you that they are not interacting. AlphaFold might do that because it's not binding them. So I think for peptide, you should look at the work of Ora Fuhrman. She has worked a lot on protein peptides. There are also Rosetta protocol there, so maybe you want to look at what they are doing. And AlphaFold has been using for peptide as well.
That's really great advice. Thank you very much, Alexandra. So Kikuti says, thank you, Alexandra, for all this work. Can you give us a rough idea of the maximum number of atoms in an all-atom HADDOCK 3 simulation?
Okay, so I ran into this problem last week. I have a visiting PhD student from France, and we wanted to dock some small molecule on the ribosome. And there, okay, and we are still using the old PDB format. So if you think the old PDB format, maybe I should not say here, it's so MMC these days, but the old PDB format, the maximum number of atoms that you can represent is 99,999. But CNS, which is used as engine behind HADDOCK, can use the hybrid 36 format, and many of you will have no idea what I'm speaking of. And it means that we can go past that limit. So this, this ribosome system, we went up to 150,000, and it's fine. Remaining then becomes a computing problem, because then everything becomes very slow because you have many atoms. But many tools will struggle with those limits. There is also a limit in the number of residues that you can describe in a PDB file. So we have to play some trick to do things to get things working, because we hit this 99,999 limit. Coarse graining will help you a little bit, because then you probably saving the mapping is often 1 to 4, so you will save about 1/4 the system. But in a HADDOCK pipeline, you start all atoms, so you still have this limit. But it's a limit we can lift, but it will not lift it on the server, because we don't have infinite computational resource. So if you want to look at very large system. And the other thing to realize, is that if you know where things are binding, you don't need to represent the entire system to model that part. Okay, so only keep the parts that are relevant for what you are doing, and simplify the system for the things that you don't need. And you can always reconstruct it afterwards.
Excellent. So Giuseppe asks, how can DRIIMB be improved in your opinion?
No, we've been trying. So now the original DRIIMB was improved first by using the large language models, and then we moved into the antibody-specific version by adding new features. And we've only been working on proteins for DRIIMB so far, so we could also, move into nucleic acids as well. There, it's sometimes a bit more tricky in terms of complexes. And one thing, there is also a preprint in bioRxiv. I didn't speak about that, but you can also improve it by doing fine-tuning. So you can doing fine-tuning of your model at prediction time. So you don't need to retrain the model, but you can do this. There is this method which is called LoRa, which does fine-tuning. So we have a paper in bioRxiv, and it's now in a revision. It's called AB-Tune. And in that one, we demonstrate fine-tuning for free application, which is for structure prediction using ESNfold, for delta G prediction, so predicting impact of mutations. And also, there's a third application that I don't remember what it's about right now, but and this is all about fine-tuning. So these are things. So you can fine-tune your model. So you can take an existing model. You don't need to retrain it from scratch. Some of those models we cannot afford to retrain it, but you can fine-tune your model for your specific question, and this might improve things.
Excellent, thank you. Hewan asks, can DRIIMB score AlphaFold predicted protein DNA complexes?
No, we never train it on protein nucleic acid complexes. So that's there is not so many. So they are well, in principle, we could train a version, because the first version was trained using a HADDOCK model of about 150 complexes and generating millions of models. The version for antibodies, we train it on 1,500 models, but we never looked at protein nucleic acid yet. So in principle, it could be done, but we have not looked into that.
Tanya asks, can deep rank antibody be used to score models where the antigen is not just a single chain? For example, a peptide HLA where the antibody binds to both the peptide and the protein.
That's a good question. That's because I think now it can only deal with 2 chains at a time. So the question is, how would you do multiple chains? So probably the peptide and deep rank, well, deep rank AB, well, the antibody MHC you could score. I'm not sure if the peptide it will contribute a lot, but at this time you can only do 2 components at a time. So you could treat everything as one single protein and then hope that it will be working, but then you will have to trick, if you want to use ESM embeddings, you will have to trick to give the peptide also to the ESM, and I'm not sure how that will work and what will come out of that. So that's a tough one.
Similar question from Chandan. They ask, does AlphaFold predict the docking of 2 different ligands at a time simultaneously?
You can do that. Now, if ligand you speak small molecule or ligand you can protein, so AlphaFold multimer, but also AlphaFold 3, you can have multiple chains. So you can do that, yes.
Okay, and one more. There was one in the chat that unfortunately I just missed, but never mind. Does AlphaFold predict the docking of a ligand if the active site is present between the protein interface? I don't understand that one,
Okay,
Chandran.
so yeah, so a buried binding site or something like that. But AlphaFold does end-to-end prediction, so it's not like, when you do classical docking, you might have a binding site which is buried and not accessible, and then you have to get the ligand inside. But I guess in AlphaFold 3, it's only AlphaFold 3 that can deal with small ligands. You could do 2 proteins plus a small molecule. I'm not sure how good it's doing with for small molecule docking. There are many other methods out there. Bolts, and Bolts 2 is quite interesting now for small molecule binding affinity. Another interesting one is Proteinics. It's coming from Chinese. I think it's the research is funded by TikTok, basically. But they have a nice open source model. It's on GitHub. They have a web server. So I have not played with it, but it seems to be quite promising. So there are many methods out there. So I think you should really test and benchmark those.
That's brilliant. Thanks, Alexandra. So I think we exhausted all of the questions now. You've had quite a few. So thank you very much for your amazing talk. It's lovely to see you again.
Thank you.
Anything from you, Martin or Bindu or?
I think there's one question at the end from Rihanna. The Rihanna Lampa.
Have we got one more? Sorry, am I missing somebody? Oh, yes, there's one from Brian now. Brian says, in your pentamer channel example, how tightly did you maintain symmetry? Your magnetic equivalents could come from rapid averaging.
So we have these C5 symmetry restraints in this case, which are, and then we have the non-crystallographic symmetry as well. So we do, and if there was a strong averaging, it will also show in the relaxation rates, I think, that were measured. So in the slides, there's also, look up the paper, but there are relaxation data that are showing you also the dynamic of the system. So that should be affected if there was a strong averaging of your signals. And it will also mean if there was a strong averaging that you would have broader peaks. But I don't think that's the case. Of course, it remains a dynamical system. But so there is C5 symmetry, and it's of course dynamic in the system, but there's clearly only a single set of resonances in the NMR.
Excellent, thank you. So Bindu, have I missed anybody else?
Yeah, there is one question in the chat.
Okay.
From
Oh, I
Riano.
can't see that one. Could you read it, please?
Yes, yeah, no problem. Are solvent molecules, that is waters, that is solvation or desolvation energies taken into account during docking?
So the desolvation energy term that I described is not using explicit solvent molecule, but we have worked in the past on a solvated docking problem, because if you look at complexes, about probably a quarter of the interface will have some water molecule there. Some are drier than others. They are very wet interfaces. So we do have a solvated docking protocol that we benchmark on protein, and protein nucleic acid, where the modeling starts from a solvated protein, basically, and then you have to get rid of the water during the rigid body minimization. And we do that in a Monte Carlo process and using some probabilities of water-mediated contact. At the time, it was improving slightly the results. Now we basically never use it in the lab, but I still see people using it and telling me, yes, for their problem, it was very useful. I know that people are using that for for peptide protein docking, and they seem to be happier of the results. But yeah, so the ability is there, but we don't actively use it. And it's not in HADDOCK-3 anymore, something that I must see. So we stripped a lot of things from HADDOCK-3, cryo-EM also, sorry, Martin, but that's something we have to reintroduce. And it will be reintroduced.
Yeah, I forgive you, but it's always difficult when you try to re-engineer something.
Yeah, so start simple, and then we will make it modular. So we'll have module dealing with different things, and because before it was one big, piece of code that was doing everything, and then it becomes not manageable anymore.
Absolutely.
Sorry, one last question, like just posted. Are stacking interactions between aromatic included in the force field? All
No,
atom model.
we don't have that. It's something that I like to see when I look at model. Again, look at structures if you work with protein structures, but we don't have an explicit term to favor stacking. This will have to come from the van der Waals and the electrostatic as well, because typically on a ring, the atom, the hydrogen might have and the carbon will have slightly positive and negative charge, with my favor stacking. That's also what you see in nucleic acids. But we are thinking of having something, because in some cases it might be handy to be able to enforce a bit more stacking for some applications.
Excellent, thank you. So,
Okay.
can I just ask Bindu? So people have asked in the chat. So the recording, it will be posted on the DRIIMB website, or
the recording will be made available on CCP Biosim YouTube channel.
okay,
Yeah.
fantastic.
Yeah, so that's where all the DRIIMB webinars are going.
Fabulous.
Okay.
Brilliant. Thank you so much, Alexandra. Really nice to see you.
Yep. Thank
Yeah.
you for the invitation, and thanks for everyone who was there. It was a nice audience.
Yeah, great questions from the audience as well. So yeah, fantastic. Brilliant. Okay, guys. Well, anything else, Martin, before we go?
No, that's thank Alexandra again. That was a great
Yeah, it was great and very popular with our community. So thanks, Alexander. Thank you so much.
Bye-bye.
See you
Have a nice
soon.
Thank you.
Bye, everybody.
Thank
end of the
you.
day.
Bye.
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 Bye.